These are AP Biology notes on photosynthesis. Today we'll talk about an overview of photosynthesis and the nature of light. So the reason we need to learn about photosynthesis, of course, it is the process that feeds everything on Earth. It changes the energy, the radiant energy, electromagnetic energy of sunlight into chemical energy that is stored in food. And almost everything in the world depends on photosynthesis for life. There are a few types of organisms, mostly bacteria, that undergo a chemosynthetic process rather than photosynthetic, but otherwise the autotrophs in general that we depend upon are photosynthetic organisms. The photoautotrophs is what we call those, as opposed to chemoautotrophs, are going to use that energy to make the organic molecules that power us. So we're using sunlight. It occurs in various kinds of organisms, plants, of course, algae, certain other kinds of protists like euglena, and various other protists that have chloroplasts. And there are some prokaryotes like cyanobacteria that are also photosynthetic. There are, of course, also purple sulfur bacteria and a few others that undergo a chemosynthetic process. But most organisms are going to depend on the photosynthesis for their food. Heterotrophs have to get their organic material from other things. Okay, they're the consumers. So almost everything depends on photoautotrophs for their food and for oxygen as well. Fossil fuels as well are formed from the remains of organisms that died many years ago. So they are also stores of solar energy from the past that are stored as a result of photosynthesis. What we're doing here is we're converting light energy into chemical energy. And this happens in a structure called the chloroplast. Chloroplasts are very similar to in structure and various processes to photosynthetic bacteria as well as to mitochondria. And the way these cells are organized is what allows for these chemical reactions to occur in photosynthesis. The main location of photosynthesis is, of course, in the leaves of plants. Plants are green, or mostly green, because of the pigment chlorophyll, which is found in the chloroplasts. They're usually found in the cells of the mesophyll, which is the interior tissue of the leaf, and that is the main site of photosynthesis. Each cell contains around 3 dozen or so, 30 to 40 chloroplasts, depending on the species, the size of the cell, and so forth. As an overview, the carbon dioxide enters and oxygen exits the leaf through these openings on the leaf called stomata. They allow for gas exchange there. The chloroplast is found in those membranes. The chloroplasts are found in the mesophyll cells, and within the chloroplast we have the thylakoid membranes, which are the stacks of these sac-like membranes, stacked into stacks called grana. They're connected to each other, and they are the site of those protein complexes that perform this magical process. The chloroplast also contains the stroma, which is a fluid inside the center of the chloroplast, in between the thylakoid membranes. Here we see a closer view of the same thing, showing the stacks of thylakoid membranes. The stroma is the fluid around them, and they're all located inside the double membranes of the chloroplast. Now, when we look at these reactions, similar to what we saw in cellular respiration, okay, we've got the use of carbon dioxide and water plus the light energy to produce sugars like glucose and oxygen. Notice the equation here shows 12 hot waters going in and 6 waters coming out. Normally when you see the equation for photosynthesis, it only has 6 waters because it shows the reduced form of the equation. But there's water used in the first part and also produced in the final part. The chloroplasts are going to split the water into hydrogen and oxygen, and they're going to incorporate those electrons into different molecules. And you can see here where the atoms go from each of the reactants into the products. It's basically a redox process, just like we talked about with cellular respiration. and it basically is reversing the direction of electron flow as compared to what we saw in respiration. So oxygen, water is oxidized and carbon dioxide is reduced in this process. There are two main stages of photosynthesis. We're going to look at an overview, a kind of a preview of what happens and in the overall process, and then we'll spend more time talking about the individual parts of the process. The two parts are called the light reactions and the Calvin cycle. The light reactions are the photo part of the word, and the Calvin cycle is the synthesis part. The Calvin cycle is also sometimes called the light independent part, or the light independent reactions, sometimes called the dark reactions. But the term dark seems to indicate that it happens in the dark, and so we don't usually use that. We usually use either the Calvin cycle or the light independent phase. The light reactions in the thylakoid membranes do these things. They split the water, releasing oxygen. They reduce NADP plus to NADPH, and they make ATP from ADP by photophosphorylation, a different type of phosphorylation than we talked about before. The Calvin cycle is going to then use the ATP and the NADPH produced from the light-dependent reactions to form the sugar from carbon dioxide. And it's going to begin with carbon fixation. It's going to incorporate carbon dioxide into organic molecules step by step, producing as a result a 3-carbon compound that is the basis of the sugar. Overview diagrammatically here, we see the chloroplast. Light strikes the thylakoid membranes in the chloroplast, uses water, splits water, releases oxygen, reduces NADP plus and ADP to produce ATP and NADPH, an electron carrier similar to NADH that we saw in respiration. Those molecules are used in the Calvin cycle to change the carbon dioxide into the sugar. And here we see just the generic carbohydrate equation rather than, or formula rather than the longer glucose, because the ultimate product of this process is not really glucose but is G3P, which is a 3-carbon compound. The light reactions are going to convert the solar energy into chemical energy in the form of ATP and NADPH. And they're going to do this by a similar process to what we saw at the end of cellular respiration, and that is by means of an electron transport system. So since we're talking about light, we have to talk light energy, we need to talk a little bit about light. Light is electromagnetic energy, and it travels in waves, but it's also called photons. Okay, the wavelength is going to determine the type of energy that is found in the energy, and in the terms of light, visible light, the wavelength is going to determine the color of the light that we are able to see. Remember, when we talk about the electromagnetic spectrum, we're talking about the whole thing from radio waves and microwaves all the way to ultraviolet and cosmic rays. So visible light is what we're talking about, the wavelengths that produce colors we can see. Realizing, of course, also that light behaves as though it consists of particles called photons. And so we'll talk about photons and wavelength kind of interchangeably here. Here we have a diagram showing the entire of the electromagnetic spectrum, from the very long radio waves to the very short gamma rays. Okay, more energy in the shorter wavelengths, less energy in the longer wavelengths. And right here in this part here is the part that we call visible light. The wavelengths of visible light range from 380 nanometers to 750 nanometers. 380 for the purple. Below that is ultraviolet. 750 for the red. Above that is the infrared. So remember, shorter wavelengths have higher energy, longer wavelengths have lower energy. Now, the pigments in photosynthesis are the receptors of light. Okay, they are substances that absorb light. Remember that different pigments absorb different wavelengths of light, and the wavelengths that are not absorbed are either reflected or transmitted, and that's the color that we see. So leaves are green because the chlorophyll reflects and transmits green light, and absorbs other colors. Here we see the main colors that are absorbed are in the blue, violet, and the red, orange range. And the greens are reflected and transmitted. We can use an instrument called a spectrophotometer to measure the ability to absorb wavelengths by sending light through pigments and measuring the fraction transmitted. And so this just diagrammatically shows what happens with a spectrophotometer to show that the transmittance of the different wavelengths through the chlorophyll. Green has high transmittance, low absorption. Blue has low transmittance and high absorption because the chlorophyll absorbs most of the blue light. The absorption spectrum shows a graph that plots the light absorption, okay? And so we can compare the absorption spectrum of chlorophyll A and chlorophyll B to see what's going to work the best for chlorophyll, for photosynthesis. Here we see an absorption spectrum for chlorophyll A, chlorophyll B, and accessory pigments called carotenoids, okay? Notice that we have peaks here for both these chlorophylls in the violet-blue range, and again in the red-orange range, but very little through the greens and the yellows. The action spectrum is going to measure the rate of photosynthesis under different wavelengths of light. And so here you see that you have a lot of photosynthesis occurring in this region of the spectrum and in this region, but very little at all in this region. Back in the 1880s, a scientist named Theodor Engelmann did an experiment to try to show what wavelengths of light were more significant for photosynthesis. He took a filamentous algae like Spirogyra and exposed it to, put it in water that had been depleted of oxygen, and included some aerobic bacteria in the water, exposed that to the spectrum that had been separated out by a prism, and let it photosynthesize for a while. He found that the photosynthetic bacteria gathered in the areas where active photosynthesis was occurring because that's where oxygen was being produced and they needed oxygen. And if you look at the pattern here formed by the bacteria, you'll see that it very closely mirrors the action spectrum of chlorophyll, of the various chlorophylls, and the rate of photosynthesis. This was done in 1883, and he used that clustered along the algae to measure the bacteria clustered along the algae to measure the oxygen production, which indicated that more photosynthesis was occurring there because more oxygen was being produced. Chlorophyll A is the main photosynthetic pigment. There are also chlorophyll B that can be used, and that lets the photosynthetic process use more light. And then there are other accessory pigments like carotenoids that absorb excessive light, kind of like sunscreen does, to help prevent damage to the chlorophyll molecules. Here we see a generic chlorophyll molecule. It could be either chlorophyll A with the methyl group as shown here, or chlorophyll B if you had this group up here, but basically the same thing. You've got your porphyrin ring with magnesium in the center and a hydrocarbon tail. Now when the pigments absorb light, what happens is it changes the ground state to an excited state. The electrons get excited and they move to a higher energy state, and then as they fall back to the ground state, they give off photons, and that causes something called fluorescence. So when you look at the solution of chlorophyll, when you eliminate it, especially with ultraviolet light, you'll see that the photons get pushed up to, the electrons get pushed up to a high excited state, and that causes the fluorescence to give off, the photons to give off fluorescence as well as heat. And here you see the chlorophyll solution that is fluorescing in the red range.